Topic 7.13, the origin of life. This is one of the coolest topics in biology. For AP biology, you need to master a very specific body of knowledge. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. The key question to address when trying to understand the origin of life is, how did life emerge naturally? And this connects to some biology that you've already learned in your AP biology course. For example, you've learned about the cell theory and the idea that all cells come from pre-existing cells. Well, at some point there had to be a first cell, and in fact, that moment really marks the emergence of life. How did a cell emerge with the absence of another cell to create it? That's one of the mysteries that we have to contend with in the origin of life. The other one has to do with chemistry. The chemistry of life is largely controlled by complex proteins called enzymes. Enzymes can take simpler substances and make them into more complex substances through processes like dehydration and synthesis. Well, how do we get the complex substances that life is based on in the absence of enzymes? What are the key steps required to explain the origin of life? The Earth is extremely ancient. It's been around for 4.5 billion years. And life is thought to have emerged about 3.8 billion years. What steps allowed life to emerge? The first thing is that Earth needed to become a habitable and more stable planet. The Earth formed from a nebula about 4.5 billion years ago. That's beyond the scope of this course. But what's important to know is that for the first several hundred million years of Earth's existence, it was pummeled by asteroids and comets, and all of that needed to come to an end before life could begin to emerge. Step number 2 is about chemistry leading to biology. Biology is based on cells, and those cells in terms are based on complex molecules, polymers. The polymers are built from monomers. So how did those monomers emerge? That had to happen abiotically. In the absence of enzymes, monomers had to start to emerge. We need to start to have amino acids, nucleotides, fatty acids, etc. In today's living world, monomers get combined into polymers. How could that happen in the absence of enzymes? There needs to be some kind of abiotic process by which that could happen, where we have the abiotic synthesis of polymers from monomers and the formation of vesicles. Those are going to become the little capsules into which cells will emerge. In step 4, we need to combine those monomers and polymers into those little vesicles to create protocells. These aren't quite living cells the way that we have them today, but they have many of the characteristics of cells. And finally, we have the emergence of self-replicating cells. That would be the last universal common ancestor over here, and that gives rise to the 3 great domains, archaea, bacteria. Those two combine, that's another story altogether, to form the eukaryotes. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. The Miller-Urey experiment, which was carried out in the 1950s, showed that in an abiotic setup, without life, you could successfully synthesize amino acids in a simulated early Earth environment. Now, as we'll see, some of the details that were involved in the setup of the Miller-Urey experiment were wrong. They don't correspond to our current understanding of the geology of the early Earth, but it's an important proof of concept. So, what did Miller-Urey do? They created this sterile apparatus with various chambers and tubings, and the whole thing was sealed off from the outside world and sterile. This chamber over here at number 2 represented the early oceans. It was heated by a Bunsen burner or some other heating device. And then the steam circulated through these tubes over here. This large area over here is a chamber that represented the early atmosphere. And it was loaded with gases that were supposed to be present in the early atmosphere. And that includes gases like methane, ammonia, hydrogen, and water vapor. Those gases were chosen because in the 1950s, that was the current understanding of what the early Earth's atmosphere would be. There are two important points. One is that that was chosen because that was the known atmosphere of some of the gas giant planets like Jupiter, and it was thought that that might also be present on the Earth. But the most important thing was that no oxygen was present because oxygen was produced by photosynthesis. It wasn't present in the atmosphere of the early Earth. There were electrodes, shown over here at number 4, by this plus and minus sign. And what they did is they produced sparks, which simulated lightning. The gases continued to circulate. They circulated through this condenser over here, and that caused whatever was formed in this chamber here to be caught within this trap. This trap could be sampled without contaminating the apparatus. And after a certain period of time, Miller, who was the actual experimenter, it was Stanley Miller, Harold Urey was his graduate thesis advisor, sampled the liquid and they found the presence of amino acids. Well, like I said, that didn't necessarily establish that life could be produced in the absence of life, but it did establish that some of the monomers of life could be produced in the absence of life. As I said, some of the details were wrong, but this has proven to be the model for many subsequent experiments where there are inorganic catalysts and different mixes of gases that are changed, and in subsequent experiments, the nucleotide bases have been produced, fatty acids have been produced, many of the monomers of life have been produced. So, what's the basic idea? In the absence of life, it's been experimentally established that some of the monomers that make up the chemistry of life could be produced. Are you asking yourself, how am I going to get a 4 or a 5 on the AP Bio exam? It's a good question because it's a hard test. But we have a plan for your success. Go to learn-biology.com, sign up for a free trial, and complete our interactive tutorials and interactive AP Bio exam reviews. We guarantee you a 4 or a 5 on the AP Bio exam. See you on learn-biology.com. To understand the origin of life, you have to understand the origin of heredity. It's widely thought that RNA, not DNA, was the first hereditary molecule. Why? RNA does store genetic information in viruses, so it has the capability of being a genetic molecule. But unlike DNA, RNA can also act essentially as an enzyme. RNA can have catalytic properties. It does so in ribosomes, where the catalytic part of a ribosome is what actually stitches together amino acids to create polypeptides and then proteins. We have spliceosomes, we have microRNAs, all of which are catalytic, acting upon the world. DNA, with its double helical form, is fantastic for storing genetic information. RNA, because it has both catalytic abilities and information storage capabilities, is widely thought to have emerged as the first genetic molecule. Before there were cells, it's thought that there might have been a phase in which there were self-replicating systems of RNA molecules that were subject to natural selection that would grow in complexity. And that's called the RNA world that led up to the last universal common ancestor of life. Here's how that might have happened. In the beginning, what we had were inorganic precursor molecules. It's what we talked about in the preceding slide with the discussion of the Miller-Urey experiment. So at some point, the sugars, the phosphate, and the nitrogenous bases that make up RNA would have to emerge. Those would combine through abiotic, inorganic processes to form RNA monomers. Other abiotic, non-enzymatic processes would lead to the next step. And in that next step, we have RNA polymers that start to emerge. They're shown at C. RNAs can fold up into complex shapes, and that's what we start seeing over here at D. And those complex shapes can start to have enzymatic properties. One of those enzymatic properties could lead systems of RNAs. It doesn't have to be just one molecule, but there could be a system of interacting molecules that could lead the system to replicate itself. At some point, those self-replicating RNA systems would become encapsulated within some kind of lipid bilayer. And at that point, at letter F, we have a protocell. Further natural selection, further evolution would lead to the emergence of the last universal common ancestor. That's the population of organisms that gives rise to both archaea and bacteria, and ultimately through a fusion of those two domains to eukaryotes. And if you can look at this diagram, you should pause and test yourself and identify all of the numbered items. Then you've learned a lot of cellular biology and you're well on the way to being set up for success on the AP Bio exam. Just to run through, at number 1, we have a lipid bilayer. Remember, it doesn't have to be a phospholipid bilayer because archaea have a different kind of bilayer structure. At 2, we have DNA being the genetic material that's found in all living things. At 3, we have RNA, which is used for information transfer and other things. At 4, we have ribosomes, which are translating messenger RNA into protein. At 5, we have a membrane channel that's allowing matter and energy to flow in and waste to flow out. At 6, we have complex proteins that are acting as enzymes, which are combining monomers into polymers through dehydration synthesis and then pulling apart those polymers through hydrolysis. And at 7, we have ATP synthase, which is using a flow of protons across the ATP synthase channel to generate ATP, that universal energy molecule from ADP and phosphate. That's the last universal common ancestor, and that's what you need to know about the origin of life for AP Biology. Here are your next moves for AP Bio success. Please subscribe to learn-biology.com and please watch this next video.